Right, yeah.
itself, but if it's kind of all over the place. So that's
Bless you.
But I will tell you that I remember the very first time Shane tried it. That's exactly what it
did it?
did. I don't remember what he changed, but I know that's what it did first.
Yeah, there's just a bunch of settings. Well, it sucks because there's a bunch of settings that I just have to keep trying, but then each simulation takes like hours to do the sequence. So that's just basically what I'm doing. It's just putting different things and going, trying to follow that.
Why does it do that?
I can't do it. You have to stretch. I don't know why. So. Wrong.
I heard wrong. What? Did you put an announcement about the test?
No.
Okay.
But I'll just talk about it. Well, she would talk about it.
No, that's fine. Just make sure they know it's they're not gonna. They don't need to come in on Tuesday. Wait until the following Monday, which is the
9th.
9th.
All right. We're going to start. Obviously, a few announcements I'll reiterate. No test this week. You'll just have a study guide that you will finish online. And we just decided to give you through the 9th. So, since this next week is Independence Day. So you'll have through the 9th to finish that. And it's basically you just finish. You can finish the study guide. It's on Blackboard, and you'll take it once in one sitting. So don't. You can take as long as you want. If you just want to show up and run through it, you can. If you want to, if you want to spend all the time, open whatever you want. Right? Is that what you're saying? You're welcome.
No, that's fine.
Yeah. We'll give you kind of a break on this. Okay, let's just be honest. No one really likes any of this topic, but this is one of those topics that we have to do. I mean, metabolism, all of us are kind of like, okay, well, I get that. But biosynthesis of all this stuff, there are some important drug targets in here. But this is just something we all have to endure. We had to endure it, so you have to endure it. That's the way it works.
Just another quick note about tests because the finals coming up.
Yeah.
You won't get that second part that we've been doing. So this is the double-edged sword of doing tests this way, where you take it and then you get to take it as a group the second time. The double-edged sword is that when it comes to the final, you don't get the groups part. So it's completely based on your individual test performance, which means if you haven't been doing as well as you wanted to do on the tests, maybe start studying with others, come and talk to Ben or myself. Let's see if we can help you maybe change the way you're studying before the final. Okay?
It's a hard test.
And what time on the 9th is it closed at?
Oh, the study guide?
Yeah.
We'll probably just leave it open on the night on
Okay.
11:59 on the night, right?
the 9th. 11:59 on the 9th.
So you have all the way through Monday. Any questions on that? Yeah. There is a lot of solid biochemistry in the topics. So in this week's topics. So and it's a lot of just reactions and stuff. So the final will be an hour, right? Isn't that?
It has to be an hour.
It has to be an hour. So my one was like 65 minutes or something crazy. So I was validated an hour. 60 questions will be done. Then you'll have the rest of your summer to yourself. As you might remember, we don't have class next week, right? that would be a good time to get some of your video stuff done. I want to introduce Adam to you. This is Adam. He's a P2 pharmacist. And so he works for me. He's worked for me for a long time now. You're crazy. Adam knows Pinewall twice as good as I do. Because anymore I just say, Adam, hey, go figure this out. So he also works for Amanda now, doing simulations. And so
Nice to meet you, Adam.
he's a resource for you. So if you would like to, if I'm too unavailable, or you don't like my breath or something, I'm fine with that. You're not gonna hurt my feelings. You can just ask Adam. Although his breath sometimes, iffy. So yeah. So here's Adam. If you need to get a hold of him, it's just adam.cosmore@issue. I'll put that up for you. Are curious. So this today's topic is we're probably not going to fill the whole time. One of the things that I wanted to do though is I wanted to give you an example of how, and we'll just do it on the fly. And Adam's going to provide input, and Adam's going to provide input. You're welcome to provide input. Just pick a topic that maybe you're not going to do one of your videos on, but that you want to see how some of the processes that you can undergo. Does anyone have a topic they're interested in, but maybe not doing a video on?
I sent you one earlier today.
You want to do concavulin?
Well, concavulin is one, but I sent you another one too. It's an MS drug. It's an antibody-based drug. We talked about it in class, but I started looking at the structures and the way that it interacts with integrin. It's integrin 4, and I can't decipher which chain is which.
Does anyone have it? Oh, we can do both. So I'll leave it up to the class, and then we'll do yours maybe at the end.
Absolutely. If anybody has anything else.
What is there? Yeah.
I'm confused.
I'm going to give an example of how to do background research to do your videos, and I think this is I think there's value in this, in just when you guys get in and you start thinking about a drug that you have no frame of reference for. This is also something I want to give you a tool. We want to give you a tool that you can actually go and look up and maybe have an idea of how to deal with it when you from a biochemistry perspective. I understand that may not be your most favorite way of doing things, but frankly, I think that, I honestly think, because I'm a biochemist and I'm biased, I think you guys don't get enough biochemistry. In fact, there's people in the pharmacy world who think there's too much biochemistry, and they're wrong.
That's Adam. He thinks there's too much biochemistry.
And that's just plain flat wrong, I think, because frankly, hey, look, no one wants to do organic chemistry, right? I think that's the line. Let's be honest, right? But biochemistry, I think it's really, really interesting. Now, I don't know necessarily you guys need to get down to the bond level, but I think understanding the differences, the molecular structure, I think and the targets, the biology, the story, I think is important. If I were a drug dealer, pharmacist, I would definitely want to know that. I think I love it. Anything? Any disease? Any drug? A drug you heard on the news? A drug your dog is taking? Your mom? Okay.
No, Accutane.
Accutane. Okay, let's do Accutane. I don't know. Okay. I have no. Wait, this wasn't a plant. We're just going to try this out. Accutane, right? Oh, so okay. So it's a vitamin A derivative.
Are we supposed to be able to see this?
Oh, sorry.
You're supposed to be imagining.
Sorry, I have. Okay, so Accutane, vitamin A derivative, treats severe acne. So, what do we think it is? Just based on what you're just based on that. Okay, that's probably a good.
Do you think it's a fat soluble drug? Or do you think it's a water soluble drug?
Water.
You think it's fat? You think it's water? Well, this will be interesting. I don't know the answer to that. Vitamin A is fat soluble. So if it's a vitamin A mimetic, but it says vitamin A derivative, is that what it's like then?
Well, so here's the drug. I mean, that's pretty long. That's a pretty straight chain hydrocarbon, right? There's two groups that might have some polarity, right? 2 oxygen there. I'd be willing to bet that it's pretty hydrophobic, and that head right there would stick out into the membrane, and the rest of it would be fat soluble. I would, that would be my guess. All right. So here's,
Interesting question.
it is an interesting question. I'm seeing if I can find it.
What are you looking for?
So I'm gonna see if it's fat. What if it said it was fat soluble? So here's definitely some structure to it, but it's relatively flat, right? That hydrocarbon is pretty flat. Oh, it's got double bonds all the way through there. Okay.
Does that mean it's saturated or unsaturated?
All right. So let's look up. I'm not seeing anything in the PDB. So that's what you get. Oh, wait, maybe this is it. Let's see what this is. Oh, so polyamine and amino acid transporters. That's interesting. Is this a polyamine or a polycationic compound? Is there any amines in that? No. So I'm not thinking that's... So let's just type this in. Mechanism of action. Well, let's look in the Wikipedia again and see if we can see if it has the mechanism of action.
It's a no.
You picked the one that was just like totally nondescript. Actually, but that's okay. No, but it's okay. Induces apoptosis in various cells of the body. What? How would that? Why would that?
It's doing something with retinoic acid.
It says has a low affinity for retinoic acid receptors and retinoid X receptors. Agonists of RAR and RXR nuclear receptors. Neutrophil. I mean, so here's a structure, right? So that definitely would be something that you could look into. Okay, so here we go. Downregulate telomerase enzyme and H-TERT inhibiting cellular immortalization and tumorigenesis. Oh. Here's your MMP9, man. So MMP9, yeah. MMP9 is known. You want to discuss the MMP9? That's your thing, so I don't want to.
Well, MMP9 is upregulated in cancer. So it's matrix metalloprotease, which means it's a protease, so it's going to cleave a bunch of proteins. And it's known to be highly upregulated in cancer.
Oh, yeah. So it is fat soluble. So, if we were going to tell a story with this, right? I would pick one of these topics and discuss how that impacted, take one of these mechanisms of action and suggest that that's a potential mechanism of action for your story, and discuss the mechanisms and how it would bind. Some of the things that we can do is if, for instance, you take one of this retinoid X receptor and you wanted to, there's probably PDBs for this. He hopes. So here's a crystal structure of retinoic acid, binding complex domain. And what we could do, if you're really interested in the topic, we could take it to a small molecule binding. And I bet you could do the search. How many of you guys have used PubMed before? Google Scholar? I don't even use PubMed anymore, so I'm not sure they should do Google Scholar. The advantage of Google Scholar is, well, I can integrate in with my library, and I can, with our library, and I can get direct access to all the files that I want. You get everything with Google Scholar, right? And you can search by date range, but you get everything. What's the advantage of PubMed if you're searching? Anyone know? You can. Did you know Google Scholar? Who just say no? Yeah. So shut off my. The advantage of PubMed is that you get group chatting. The advantage of PubMed is that you get a very local. It's a very it's a narrower search. So if you get Google Scholar and you're like, it's like you're trying to figure, you can't see the forest from the trees, you can search in PubMed and you can get a much more refined search quickly. I think, how much did you guys use PubMed in your
Druglet, we use it all the time.
Yeah, so you'll learn how to use it in Druglet. There's no such thing. Okay, so we may do the same exercise here in a little bit just to help you out a little bit. Otherwise, we're going to start into the topic. Do you have any questions about that? On what you can do with that? I would recommend that you probably look at one, maybe two articles, journal articles, if you're going to do your video. It's not required, but it definitely gives you an idea. Oh, the other thing you should look at is, so if you were going to look at Accutane, I would do a video search. I bet if you looked at, if I type in mechanism of action, and I do a image search, there's probably going to be some good images that you can use for a video. See, I would have predicted that Accutane would have been an antibiotic.
It does have some antibiotic.
Yeah, but it looks like it inhibits sebaceous gland. I'm sure that retinoic. So the story you would put together would be, you'd find out how it reduces sebaceous glands. That retinoic receptor may reduce sebaceous gland secretion, and then you tell me, you can look and actually see where it binds and put that story together all the way down. You say, hey, look, when you get acne, you end up with all kinds of sebaceous gland secretions. To decrease that is this receptor. Here's the drug that inhibits it, and you go down that path. I don't know if that's sometimes you get crap on these searches, so you really got to be careful. All right. Any other questions on that? You guys have been the least inquisitive about this, so I'm getting nervous about where you are. Usually I have Usually I have people bugging me like crazy about this. They're camped out in my office, literally. Adam, we can't.
We're not 3D printing everything this time.
That's true. All right, Adam, you can go have dinner. If you want to have, and Adam's here almost every day working. You're welcome to bug him. If you want to. Plus, you could also pick his brain on pharmacy school.
Oh, yeah. Plenty of things.
I don't know who wants to listen to.
We'll try out DNA replication. Now, the other topic today that we're going to talk about is nucleotide biosynthesis. I think we've talked a little bit about replication before. It's basically the process by which we take DNA and duplicate it. Why is that? Where do we need to duplicate DNA? You should have this in high school biology, right? No one wants to talk today. I'm trying to get them to talk today.
What was the question?
Did you get sucked into Accutane?
I was looking for where the slide is.
Oh, you're not gonna find it. Sorry, I haven't posted them yet. I literally was making them minutes before class started. So where do we duplicate DNA?
In the nucleus?
We do. No. Okay. Great. That's not the question I was asking, but that's the literal answer. Right? I should say maybe when do we duplicate DNA? No, those are different, right?
Oh.
That's a good guess. I'm okay with that guess, right? Let's go back to that topic then. What is transcription? How does this relate into? Yes. And what is that used for? To make proteins, right? That's translation. When we take mRNA and go to protein. Transcription is we take DNA and make RNA. Replication is when we make DNA from DNA. When do we need that? Yes?
Before.
Oh, mitosis.
Okay. Yeah, mitosis is actually what I'm looking for. Right? Right? If I need to have two cells from one, I'm just going to need to replicate. Is that a trivial task? Quick search. You guys need to do this right now. How many genes are there in the human body?
23.
That's chromosomes.
Um,
That's okay. I'm liking the. So 23 chromosomes. How many genes are there in the human body? Look it up on Google right now. I don't know the answer to this question.
Wikipedia says 19,000 to 20,000.
That's probably a pretty fair guess. That's. This actually, I'm doing this on the fly, so you're. This is actually a really good TED Talk. I'm going to go in the room. I don't know either. Can you guys hear that?
In the next 16 minutes, I'm going to take you to a journey that is probably
You
the
said
biggest
16.
dream
We won't go
of
that
humanity. Understand the code of life. So for me, everything started many, many years ago when I met the first 3D printer. The concept was fascinating. A 3D printer needs three elements: a bit of information, some raw material, some energy, and it can produce any object that was not there before. I was doing physics, I was coming back home, and I realized that I actually always knew a 3D printer, and everyone does. It was my mom. So my mom takes three elements. A bit of information is between my father and my mom, in this case, raw elements and energy in the same media, that is food, and after several months produces me. And I was not existing before. So apart from the shock of my mom discovering that she was a 3D printer, I immediately got mesmerized by that piece, the first one, the information. What amount of information takes to build and assemble a human? Is it much? Is it little? How many thumb drives you can fill? Well, I was studying physics at the beginning, and I took this approximation of a human as a gigantic Lego piece, okay? So you can imagine that the building blocks are little atoms, and there is an hydrogen here, a carbon here, a nitrogen here. So in the first approximation, if I can list the number of atoms that compose a human being, I can build it. Now, you can run some numbers, and that happens to be quite an astonishing number. So the number of atoms, the file that I will save in my thumb drive to assemble a little baby, it will actually fill an entire Titanic of thumb drives, multiplied 2,000 times. This is the miracle of life. Every time you see from now on a pregnant lady, she's assembling the biggest amount of information that you will ever encounter. Forget big data, forget anything you heard of. This is the biggest amount of information that exists. But nature, fortunately, is much smarter than a young physicist. And in 4 billion years, he managed to pack this information in a small crystal we call DNA. We met it for the first time in 1950, when Rosalind Franklin, an amazing scientist, a woman, took a picture of it. But it took us more than 40 years to finally poke inside a human cell, take out this crystal, unroll it, and read it for the first time. Well, the code comes out to be a fairly simple alphabet, 4 letters, A, T, C, and G. And to build a human, you need 3 billion of them. 3 billion. How many are 3 billion? Well, we don't really make any sense as a number, right? So I was thinking how I could explain myself better about how big and enormous is this code. But there is I'm going to have some help, and the best person that is going to help me to introduce you the code is actually the first man that sequencing, Dr. Craig Venter. So welcome on stage, Dr. Craig Venter. Not the man in his flesh, but for the first time in history, this is the genome of a specific human printed page by page, letter by letter. 262,000 pages of information, 450 kilograms, shipped from United States to Canada, only thanks to Bruno Boding on stage, Lulu.com, a startup that did everything. It was an amazing feat. But this is the visual perception of what is the code of life. And now for the first time I can do something funny. I can actually poke inside it and read it. So let me take some interesting book like this one. I have an annotation, it's a fairly big book. So just to let you see what is the code of life. Thousands and thousands and thousands and millions of letters that apparently make any sense. Let's get to a specific part. Let me read it to you. AAG, AAT, ATA. Well, to you sounds mute letters, but this sequence gives the color of the eyes to Craig. Show you another part of the book. It's actually a little more complicated. Chromosome 14, book 132. As you might expect. ATT, CTT, GATT. This human is lucky, because if he will miss just 2 letters in this position, 2 letters over 3 billion, he will be condemned to a terrible disease, cystic fibrosis. We have no cure for it, we don't know how to solve it, and it's just 2 letters of difference for what we are. A wonderful book, a mighty book. A mighty book that helped me to understand and show you also something quite remarkable. Every one of you, what makes me me and you, is just about 5 million of this, half a book. For the rest, we are all absolutely identical. 500 pages is the miracle of life that you are. The rest, we all share it. So think about again when we think we are different, this is the amount that we share.
Okay. I've watched that like 10 times. I find it very interesting. I don't know if you find it as interesting, but I think the major point in that
is how efficient nature is at, because he didn't actually go into this, but not only is that just in one cell, it's in all of our cells. All of that information is in all of our cells, and it's packed into the nucleus. So if we were to try to store it digitally, he said it would take, right, on a thumb drive, the whole code would take and fill up the Titanic 200 times, right? And he had a book out there just with all the information. So there's an amazing amount of information in there, and today we're going to talk about how that gets replicated, because it gets replicated thousands of times a day. If not, I don't even know. I don't want to guess how many it is. It's a lot of times a day, and it does it accurately. Okay, because we don't. What happens if it gets copied inaccurately? Yeah, mutations, and that causes things like cancer and diseases and all kinds of other problems. So DNA, obviously, as you might recall, is a double helix, and during the process of replication, these strands are separated. Okay, and that's what happens. So here's the DNA. It's in a double strand, and then they get wrapped around and packed really tightly. Does anyone know what that protein is right there? It's histones, right? And so the DNA gets packed around those histones. Those histones get packed around each other, and we can compact and put in a lot of DNA into a very small space. During replication, the histones unbind from the DNA, and we're going to make 2 copies. It's of that same DNA from 1. So they're separated. Each strand has the original DNA and serves as a template for the other, and that's called semi-conservative replication, meaning that new helix will have a new helix, and we'll have an old helix that are put with the new one. So that's semi-conservative. Cellular proofreading and air checking mechanisms, and Amanda and I have tried to do PCR a lot in our lives. And I say try. I've done it successfully too. It's not true. And I can just tell you that when you try to do this kind of stuff with DNA, it doesn't always work very well. We end up with error messages all the time in that DNA. And when you're trying to copy it, but your cells in your body does it all the time on a regular basis. So, all right, so what we're going to do is I'm going to go over all of these major proteins that are associated with this replication. Okay? That's going to be the process of what we'll talk about today. Okay, so that DNA replication occurs during S phase, which is one part of the interphase process. It takes during S phase of the cell cycle. Happens obviously in the nucleus, in eukaryotes, because that's where the DNA is, and in prokaryotes, it will happen in the cell. What ends up happening is there's the 2 strands. They'll open up at
an origin of replication, often called the ORI, ORI, ORIC, and we end up with these bubbles that are formed, right? If you recall back here, this is attached at the other side as it comes through, and so you end up with these bubbles that form around this polymerase. And what that looks like in real life is this. You have a DNA strand, and this is the replication bubble where you see we have strands of DNA that are being formed. This is called the replication fork. Pretty important concept. You will want to bone up on your 5' and 3' terminology. The replication fork happens at the point at which we dehybridize that DNA, and the two strands open up. So, and we're going to start adding in and copying each one of these. So, this is an optical illusion to me, because this top one doesn't look like it has near as many. But if you look at it, they actually line up. I spent like every time I look at it, I'm like, that's not right. There's something wrong with that. All right. So the first enzyme that's important is the topoisomerases, and these are enzymes that that unwind or overwind the DNA. So DNA is obviously a coil. You can imagine like a rubber band, okay? And if I'm trying to make a bubble in there, what would happen if I try to open that up? What kind of forces am I going to have on that? I'm try to wind a rubber band together and then try to open it up. What happens?
You wind it to infinity. What eventually is going to happen? You just keep winding it. Or with a hair tie. It can snap. Yep. So that's one thing that can happen. What else happens? Like, so you wind it up and then you put your finger through one side of it. What happens to the rest of the coils? Do they stay in place or do they slide? They slide, right? So that's going to overwind the head of where that bubble is. So that's what happens to a rubber band. You start turning it, and you can only coil it so much. And then if you put something in there, then it scrunches together the rest of the coils.
It also it increases the torque on that spot, and eventually what would happen is that torque would get so strong, right, that you would not be able to go any farther. The enzyme would snap. So what topoisomerase does is an enzyme. It actually cleaves the phosphate backbone on either one or both of the strands and unwinds it so that it doesn't create that torque. That's what topoisomerase does. Now, it's also known as DNA gyrase. So you'll see that because, again, we're biochemists and we like to confuse you. And there's an associated what's called a linking number, which is how much this is the extent of the DNA unwinding. So and that allows it, again, to unwind or overwind. It can overwind as well in this situation. Topoisomerases are used. There's actually quite a few topoisomerases that that exist. We're not going to go into any detail on them, but just know that there's various obviously for replication that they exist, and for transcription they exist. Because in transcription you also have to unwind. I think we've introduced at that point as well. Fluoroquinolones, antibiotics. But bind to topoisomerases and the spot and prevents the ability of the topoisomerases to function. They
bind at C6 and or C7s. So you can see them in this nice graphic right here. This is the reason I don't like black. Adam likes black. I can't stand black because it's kind of hard to see. But this is the double-stranded DNA. They bind it on the C7 right there, or C6 or C7 of these molecules, and that allows it to, then its mechanism of action is there. So things like ciprofloxacin. What's the trade name for ciprofloxacin? Cipro. Okay, so Cipro is an antibiotic that works in this way. And this is an example of one. There's quite a few antibiotics that, and one of the key features is that fluorine residue. That fluorine residue is what really binds, creates that bond. All right. So a couple other things that kind of happen as we're unwinding this thing. Obviously, we got our topoisomerase going up ahead, reducing the torque. We also have a helicase sitting right here. Talk about that in a minute. And we also have single-stranded DNA. They're called SSBs. So these prevent the two DNA strands from rehybridizing. So if we don't have these single-stranded binding proteins, well, these prevent the hydrogen bonds from reforming, which will then, if it did that, it would block the ability to form a template. All right. The enzyme helicase, this is the crystal structure. Obviously, it's from the molecule of the month. Unwinds and separates the two DNA strands. It breaks the hydrogen bonds, and it does this using ATP. They are a motor protein, so they click down the DNA strand, okay? And that's what this kind of structure shows right here. They separate these two and break those bonds. Approximately 1% of eukaryotic genes are for helicases. There's 95 non-redundant helicases in humans, and 64 RNA helicases, or 64 of those are RNA helicases, and 31 are DNA, which I thought was pretty interesting considering I can only think of 3 or 4 things that actually require, especially RNA. There's something else going on. We live in an RNA world and we don't really understand why, or what. Because the fact that we have 64 RNA helicases is really, really intriguing. And tons of cellular processes use the helicases. All right. Next enzyme we're going to discuss is a primase. So we've already talked about, see the reference here, DNA gyrase, also known as topoisomerase. Here's our helicase, separates the DNA. Here's our single-stranded binding proteins. We also have our DNA polymerases, which we'll talk about here. This is Pol3. DNA Pol3 is the one you're going to want to know about. That's the one that actually works for most replications. DNA as far as this is the workhorse polymerase. There's other polymerases. DNA Pol3 is the main one that does. We're not going to get into what the other 3 polymerases do. DNA Pol1 removes this primer, but there's 3 other classes. The only one that we care that you know about is this DNA Pol3. That's going to extend the chain, meaning, you want to take a guess what that means? No guesses? What? Oh, that's a good one. This is an Okazaki fragment, yes. That's a great, but you're getting ahead. You're way ahead of me. So, but I appreciate the fact that you're way ahead of me. Actually, all it does is it extends the chain. It's just going to keep adding nucleotides, right? Just like we did with amino acids, right? Just like we do transcription. I'll talk about this in a minute. The nucleotide's going to come in there, bind. Polymerase is going to come in and do the next thing. Let's talk about the primase. The primase actually is associated with the primer. So it's a type of RNA polymerase. It catalyzes short RNA, sometimes DNA, but mainly RNA segment. That's the primer, because DNA can't start by itself. So there's an RNA component that binds to the single strand of DNA, and the primase actually allows the polymerase to start synthesizing the strand. So this chunk of primase is actually removed by a exonuclease that's usually associated with the DNA polymerase. The next enzyme that's important is a ligase. Okay. Anyone know what a ligase does? We'll take a guess. It does. Am I killing you with stuff you've already covered in the rest of your life? DNA is one of those topics that high school biologists have a tendency to know very well. And so if you guys have seen this before, that's fine. We'll cover it maybe in a little more depth. So ligase, here's the reaction that's going to occur. You have a terminal amine. Okay, ATP is going to come in and bind here. So we're going to put a phosphate group, and we're also going to adenylate it. So adenosine is going to bind. So the two phosphate groups are going to be popped off. Right? We have adenosine with this phosphate bound to that terminal amine. The DNA is going to come in, and we're going to add in that DNA. Right? So here's the fragment. And we're going to have to create the phosphate link. We're going to start to create the phosphate linkage here, right? We're going to have, what kind of reaction is that going to be? Is there water coming out of that? Oxidative reduction? Or are there electrons going in there? It's a nucleophilic attack, right? So you're going to end up with forming this phosphate backbone, right? What kind of bond is formed at the end? Right here. So right here. What kind of bond is on the DNA? It's a phosphodiester, right? We have two ester groups, right? There's an oxygen. It's going to attach to, right? It's a phospho. Here's the phosphorus, and there's two ester bonds right there. Right? It's a phosphodiester bond. So that's all the nucleophilic attack, and it's going to go through, and that's the mechanism of action by which that enzyme, which is a DNA ligase, is going to form that phosphodiester bond. All right, so let's start putting all the pieces together. First step, helicase. Actually, the first step really is, right, we all know that topoisomerase comes in, helicase comes in, unwinds the double helix, single-stranded binding proteins bind to stabilize to make sure what doesn't happen. Hydrogen bonding. So we break the hydrogen bonds between the two, so the DNAs do not be hybridized. The leading strand is synthesized by continuous polymerization, right? So this it occurs in a 5' to 3' direction. DNA polymerase, the lagging strand, right? That's this strand. Okazaki fragments synthesize these discontinuously, right? So there's fragments that are put in there, right? And then the polymerase comes by, and the primase synthesizes these short RNA primers, and that extended by the DNA polymerase, we get an Okazaki fragment. After the RNA primer is replaced by the DNA, then the ligase comes back in and puts it all together, and we end up with two strands. this kind of shows the structure. Here's the polymerase. Here's a 3 to 5' exonuclease domain. That's going to cleave off the RNA, and then you can see all the alpha helices and how they interact with major and minor grooves, and that allows those reactions to come in that phosphate bond to be put together. This kind of gives you another cartoon that shows how all this kind of fits together. So we have a 3 to 5' exonuclease at the bottom of the polymerase. We have a polymerase domain that sits in there, and that's what's going to add and create the phosphodiester backbone. So some of the important things that are going to need to happen in here, right? You need to have magnesium in this process. That's one of the important cofactors in this enzyme. In addition, this polymerase also has mismatch repair. So what ends up happening is a nucleotide comes in and hybridizes with its complementary base pair, and that allows then the bond to form. If you have a mismatch, which means that the hybridization occurs, but it shouldn't, these polymerases have a mismatch repair that can actually go in and fix that problem. This is a different representation of what we described earlier. And then they describe this as a hand like this. So you have your thumb here, 2 alpha helices, a palm, that's basically you can see there's some beta sheets that sit in there. Then we have another finger, and that kind of wraps around the whole DNA structure. And at the bottom here is where your exonuclease will come in and clean up those RNA fragments. This is for your information. As I mentioned earlier, the important one is polymerase 3. That's the main one that does most of the work here. Polymerase 1, prime removal and DNA repair. Polymerase 2 is also a repair. Polymerase 4 is repair. Polymerase 5 is repair. So these actually have specific job repairs, so you can see that there's multiple polymerases, and a lot of them are in there just to make sure that the base pairs are hybridized correctly. What's the error rate on Pol 3?
Species dependent. Depends how much species it comes out of.
I want you guys to look that up real quick. Look up the error rate on DNA polymerase and see what you find in Google, on the interweb. So that's per base pair. So it only makes a mistake once every, what's 10 to the minus 8? It's like,
Well, 10 to the minus 9 is an animal.
yeah. So you're talking, it only makes a mistake every, but is that still a problem? That's still a problem, if you want to know the truth. That's why you have this many polymerases that are in there for mismatch repair. All right. Here is the linkage again. I think this gives a little bit better picture of your this. I think we covered DNA before we covered carbohydrates, and I think it's kind of cool to now go back and look and see what we have here. So we have our sugar residue here, right? Here's our anomeric carbon, 1, 2, 3. So that's where the third carbon is where we're linking our phosphodiester bond here. Right? And that's going to go 3'. That's where the 3' comes in. It's that third carbon. We also have our 5' position. So 1, 2, 3, 4, 5. And that's actually not part of the ring structure. That's part of the functional group that sticks out above. and that's where we get the 3' to 5' connection. This cartoon is actually somewhat inaccurate because this bond length is not the same as this bond length. There's actually a very specific bond length that actually occurs there. But
I just think it's funny that you would notice the inaccuracy.
no, if you try to model that in real life, I tried to make a cartoon representation of this, and the bond lengths are pretty important. I'll just say that. Because what other bonds do we have in DNA? We have hydrogen between, right? So an A and a T would hydrogen bond. So we would have oxygen, nitrogen, oxygen. We have nitrogen, nitrogen, and this nitrogen. So those three go together. Is that right? Anyway, we have the hydrogen bonds. What other bonds do we have?
What's the bond that connects the sugar residue to the base? What? It did. It's a beta. Yeah, good job.
Yeah, we have a glycosidic bond right here, right? So here's our sugar. It's a beta. Why is it a beta? It goes up, right? If it was down, it'd be an alpha. It's a beta. Which number is this? Yeah, so it's a beta 1 glycosidic bond, right? We have our base here, right? So we have different bases that can hydrogen bond. What other bonds do we have besides the N-glycosidic bonds? So we have our phosphodiester bonds. We have our hydrogen bonds. We have our N-glycosidic bonds. What other bonds do we have in DNA? There's one more that I'm thinking of. It happens between ring structures.
And it may have something to do with delicious dessert.
I'm not getting that one. Delicious dessert. Oh, okay. Now I'm getting it. There's base stacking, right? So these sit on top of each other. She's thinking of pi bonds.
Come on, you guys. Being super clever.
Took me longer to get the joke than I did. Anyway, so these sit on top of each other. That also helps stabilize it. It's not as much as some of the other bonds, but it's definitely there.
That sometimes means pi stack,
Yes, that's another.
which is why I. Never mind. There's also covalent bonds. Don't forget about the covalent bonds.
This kind of gives you just a little bit a more concise representation of how these things come in, form between the different base pairs. So this is added, and then the 3' is exposed. So then the 5' of the nucleotide is going to come in and bind that. So that's what 3' and 5'. Or actually considered 5' and 3'. So that's how it's going to be added in there. So the specificity of replication is based on hydrogen bonds, right? So this A and the T. So what happens if a C comes in there? It's not going to hydrogen bond, right? And so then this phosphodiester bond is not going to be able to be formed, and that precludes the ability to continue. Now that does happen, right? But the bonding isn't accurate there. It's the polymerase, one of the other polymerases is going to come in, and it's going to do excision. It's actually going to pluck that out. If this came in as a C instead of an A, it's going to go in there and it's going to pluck that out and put the right nucleotide in there. So there's an analog to a nucleotide that has shape complementarity, meaning it has pretty much the same shape, right? You can see that the red part here, we have a carbon, where this is a nitrogen, has a carbon and nitrogen. These they have a similar shape. This is adenosine, this is an analog, but it can't form hydrogen bonds because why? Why can't it form hydrogen bonds? That's what it's saying. It's saying actually that these have the same shape, right? That these two structures have the same shape. Is that completely true? No, because nitrogen to carbon is going to have a different bond length than carbon to carbon. But it's going to be pretty close, right? Considering you're probably talking, very small distances. What if this is a nitrogen? It can form hydrogen bonds, right? Can this carbon form hydrogen bonds? Why? It needs to be nitrogen or oxygen. What's the theory behind that? Has to do with the electronegativity of those molecules, right? Is this a polar bond or a non-polar bond? Non-polar, right? This is a polar bond, right? The nitrogen has more electronegativity, so it takes more of the electrons. Carbon, hydrogen have the same electronegativity, right? So that allows, this allows, when we incorporate this analog in, it actually puts in a thymidine in the DNA replication. So it allows it to, and we can use these for shape complementarity. We can use these as drug analogs. We can also use them as, in order to create missense mutations and all kinds of other things that we can use for other applications. So this is kind of like a bit of a biotech. It also shows a biotech application. It also shows how important it is when we talk about structure and function. It also shows how important it is to have the right functional groups in the right places in order for all of this to come together. DNA polymerase can only add to the 3' end of DNA, so this causes the new strand to be built 5' to 3' direction. That's pretty important to know, and that's the reason we have Okazaki fragments.
But you can only add to the what? 5 to the 3.
To the 3'. It goes 5' to 3'. And this does take, DNA polymerase takes two high energy phosphate bonds to work, right? So in order for it to occur, so 2 phosphate, and it's what kind of reaction is this? Yes, so it uses the water and uses 2 phosphate in order to create that backbone. Actually, I think I want to go here first, and I'll jump back. There are 3 steps to DNA replication, 3 general steps. They're the same general steps that you have with transcription. Does anyone remember what that was? That's been like 6 weeks or so ago now, maybe 7. Yes, that's the middle one, right? That's step 2. That's part of elongation.
Yeah,
Initiation,
that's the first one.
that's the first step. And termination, right? All 3 are the same. So we have initiation, which is where all of our proteins come together and form the replication bubble or the replication fork, right? Then we have the polymerase doing all the work, right? That's the what step? Elongation, and then it kicks it off. That's termination, right? This kind of gives, this is the polymerase, kind of gives you a nice little representation of how the elongation complex works and how the DNA sits in there, and kind of gives you an idea. It's very similar to what happens when we have the ribosome doing its work, and it allows the enzyme to breathe, and those reactions then cause it to kick out and ratchet up just like a motor down as it does an elongation step.
I will add this diagram, just like I did in the others, on transcription and translation. So just know what each one of these is. Okay. Topoisomerase, helicase, single-stranded binding proteins, polymerase. That's misspelled. Okazaki fragments are primases. Again, this talks about the conformational change that occurs as we're inside the protein or inside the nucleus. Inside the protein will change conformations as it ratchets it down based on the reaction that occurs. And it's a very, very small part that actually interacts with these. This kind of shows how these, like alpha helices, kind of shows a hydrogen bond in there, and then that hydrogen bond interacts with the rest of the DNA strand, and that actually allows it to add in to those. It's a very, very small part of the protein. It's alpha helices. That's the thumb, and then here's the palm. And those palms kind of have the rest of the DNA sitting on there inside of them, and then there's, as a protein, the enzyme changes conformation. It allows these molecules to come together, and that change in the conformation of the protein, as it allows that phosphodiester bond to occur. If you're really interested in that, there's actually two really good PDB structures on that, and I'm happy to show it to you. A couple other interesting points. This is where magnesium is green. So there's a coordinate reaction that occurs, coordinate metal reaction that occurs with that magnesium. And that's why magnesium is really important. Also, why magnesium is important in your diet. A lot of these enzymes require that. Okay, just went over that. Let's take 5. And then we'll come back and we'll end the day on a high note with nucleic acid synthesis. Is this somebody's?
All right, so let's get into how we make nucleotides. This is very enzyme and pathway heavy. The general idea is if we need to make a nucleotide, we have two pathways. We can do de novo synthesis, right, where we make these structures that are converted into ATP, GTP, and all the other nucleotides, or we can salvage them. We go down the salvage pathway, and we'll talk about what that means and how we get there when we talk about salvaging. So we can make them from scratch, or we can salvage them from the environment in different forms. We also have the ability to convert nucleotides to deoxynucleotides, so we can take RNA and convert it to DNA, and that allows us to quickly get to DNA. In this 457-step process, we can make I don't expect you to memorize this, but you might want this as a reference for your. And I kind of actually find it interesting because we start with ribose, and we start with ribose 5-phosphate, and we can take PRP synthase, and we're going to make our PRP-P, which is basically a pyrophosphate with a ribose on it. We have 3 phosphate groups in there. From there, we can take glutamine and glutamate, or glutamine, sorry, and make glutamate, and we basically take and we're going to put an amine group on our sugar, on our ribose. From there, we can take glycine, the full glycine molecule, okay, and we can attach it to this amine group. So you'll now notice that we've taken a full glycine molecule and added it to this ribose, and it's just sitting up there. From there, we're going to take methylfolate. Methylfolate, we're going to attach this formal group right here. From there, we're going to take and we're going to add and complete the ring structure in 2 steps. So now we've made the first, as you might note, our purines have 2 rings, right? So we've made our first ring structure by step 6. 6 steps, 6 enzymes. Next step, we take carbon dioxide, right? We start building up the second group. Then we add aspartate in a condensation reaction, right? We're still not, we've got this huge amino acid sticking off there. We're still not done. We split off fumarate from there, and we end up with 2 amine groups here on the purine ring. Then we convert with formal transferase. We add a formal group to that amine group. And the final step is we convert the structure here to a closed ring structure. Simple, yet effective, right? Ironically, that's step one. Step two, so at the end of this last step, we end up with IMP, right? This is the inosine monophosphate. So we have one, along the way here, we converted this ribose, this carbohydrate, this R5P, which is a phosphate group attached to the ribose at the fifth position. That's what this R5P means. So this molecule is actually this purine ring with this ribose sugar attached to it. So step two is we start with the product of that last cycle. So step two is we're going to add aspartate to it, right? So we have this amino acid stuck up here, right? And then we're going to cleave off and have fumarate come off, and we've basically added the amine to this, and now we end up with AMP. That's the next step in this process. That's how we make AMP. To make GMP, we take the same IMP. We're going to add our NADH. It's going to be converted to NAD plus is going to be converted to NADH, right? And that's going to oxidize that carbon. And then from there, we're going to take and we're going to convert that carboxyl group into an amine group, and that gives us our GMP. Any questions on that? So, here's how it's like going to the scrapyard, right? It's like those robot wars where you like, hey, go to the junkyard and build a robot. That's what the purine ring looks like. We're going to take formate group. Here's aspartic acid. Here's our carbonate, glycine, formate, and a glutamine. And we're going to like just keep adding them together, different functional groups until we get this purine ring. And they all have different locations or different sources of for each component. Here's the regulatory structure of how this works. We don't necessarily need to make a lot of DNA because we can scavenge a lot of it, but so it's pretty heavily regulated. And at each step, there's the nucleotides basically serve as the element, or IMP serves as the element for regulation. All right, so the purine ring is actually a little bit harder than the pyrimidine ring, since the purine is a little more complex. There's 6 steps. So we're going to synthesis of carbamoyl phosphate, which is what you see up here. Right. So we take CO2 and glutamine and ATP, and we can make carbamoyl phosphatate. We add aspartic acid, and we're going to end up with carbamoyl aspartic acid. And you don't need to memorize all these enzymes or anything. We're basically then going to complete the ring structure using this dihydroorotase. From there, okay, we're going to have a dehydration reaction that's actually going to create this double bond. From there, we're going to have add back in our ribose, and basically all the way through down to where we're going to end up with our final structures here. We're going to keep adding and moving around function groups until we get from CTP to TMP. And as you can see, this one is pretty straightforward. Here's kind of a little bit different representation of that same process that gives you just a. I think both of these pathways have a little bit of unique information that they present. So this one's not as complicated. Pyrimidine has a CO2. This part is aspartate, and this part is glutamine. That nitrogen, this part is glutamine. So we basically rip off the glutamine to make glutamate, and we end up with that nitrogen right there. Again, for a same regulation type thing, carbamoyl phosphate synthetase 2 is inhibited by the pyrimidine or triphosphates, so you end up with pretty straightforward regulation here. Basically self-regulation. So that's the de novo synthesis. We also have salvage pathways that can be, so we can take, and if we need to clean things up, so we can go from ribose and we can basically do the same pathway, just salvaging them out in order to and recycle nucleotides that we need. So if we have the adenine, we can go to AMP. We have so hypoxy xanthine. We can go to IMP. From IMP, we can go down the same pathway to get. We basically can recycle all of these different nucleotides. Here's all the different ways you can enter into the salvage pathway and end up with. And I don't expect you to memorize this. You can just use this as a reference again. And I think there's a couple things to note. There's zinc and magnesium as cofactors in here, as well as calcium, and these things are really important in this synthesis pathway. Last thing I'm going to talk about is we can convert RNA to DNA using ribonucleotide reductase or NRDZ or NRDEF, and basically, as you might recall, ribonucleic acid has a hydroxyl group at this 2' position, whereas deoxyribose does not. And so we can salvage that hydroxyl group out of that process from right there. And that's it for today. I'm not going to argue with you. It's one of the topics that we have.